Sampling principles. Introduction to sampling. Sampling is the process of selecting a part of something with the intent of showing the quality, style, or nature of the whole. We sample because we want to learn about the population or the area of interest. Also, it's very difficult to accurately census a population. It's often difficult to know that we have accurately measured all of the individuals in a population, and so we sample. We also sample to make inferences about the population and to generalize with confidence. Populations are the complete set of individuals or the entire area that we want to describe or make inferences about. In this sense, we're talking about a statistical population, which is different from a biological population. In the statistical sense, we're looking at the set of variables that are inclusive of the sampling universe, and so it consists of all possible sampling units. The sample is merely a subset of the population, and so it is a subset of sampling units. Our choice of sampling unit depends on the objectives for sampling, so we must always consider what do we want to know and why. This will greatly influence our choice of sampling unit so that we ensure that by doing this sampling, it's going to help us to answer the questions. In natural resources management, we are interested in different types of potential sampling units: points, transects, quadrats or plots, and individuals. In the case of vegetation, we may be considering whole plants as individuals or plant parts such as stems, tillers, inflorescences, or fruits. A variable is a characteristic that can vary from one entity to another. It can be measured or estimated, and variables are either quantitative or qualitative. Data collection is the process of gathering information about variables of interest, and we need to use care when we're selecting the variables to be measured. As Krebs said, not everything that can be measured should be. The most commonly measured vegetation attributes are density, frequency, cover, biomass, height, and age, lifespan, etc. are often collected. When we're describing populations, we use population parameters. These are descriptive measures which characterize a population. They are assumed to be fixed, but unknown and unknowable. They change only if the population changes, and we use Greek letters to denote population parameters. Sample statistics mirror the population parameters. They are descriptive measures that characterize the sample. We use sample statistics to estimate population parameters, and they will vary from sample to sample. They also vary if the population changes. We use Roman letters to denote sample statistics. Now, when we're trying to describe our populations and samples, we use parameters and statistics, mostly focusing on central tendency and variability. With central tendency, we're interested in what is the most common value. So the mean, or the average of all value of all observations, the median, which is the value of the central observation if all of the observations are ranked, and then the mode, which is the value of that observation that occurs most frequently. We're also interested in variability. So how much do these values vary? We use the standard deviation, standard error, variance, and coefficient of variation to describe variability. So let's take a look at this in a little more detail by focusing on the group of individuals that you see in the picture below. This is a marching band of 161 individuals that are ranked according to their height from 5'0" to 6'5", and they are grouped also by gender, so men are in blue and women are in white. Let's take a look at central tendency. If we look at these three different groups, we can see that there is some variation in the mean value. The women tend to be shorter on average than the men, and the group mean is intermediate between those two. The median value, which is the middle value of all that were observed, is shown here. And the median value is most often used if there's the case of some extreme outliers, because it is more robust to extreme outliers. The mode is the value that is observed most often, and we can see that the individuals who are 5'6" or 66 inches are the most often observed for both the group as well as for the women. The mean is the value that we tend to focus on when we are reporting the results of sampling. For variability, there's a number of ways of looking at this. The standard deviation is looking at the amount of spread in the values that are observed. The standard error is the spread of the values divided by the square root of n. And even though the standard deviation is different for these different groups, so is the sample size. And so at this level of precision, the standard error is the same for all three groups. Variance is not often used when we're describing populations because the units of variance are squared, and so then they do not match the units of the mean. And the coefficient of variation is a description of variability or dispersion that we use when the magnitude of the means is very different. So if we were comparing groups that had extremely different means, the standard deviations could be different in size. And so with the coefficient of variation, we relativize the amount of variability relative to the value of the mean. Now let's consider accuracy and precision. Accuracy is the closeness of a measured value to its true value. Precision is the closeness of repeated measurements to each other. It's convenient to envision this by looking at a target and assuming that the central value, the bullseye of the target, is the true mean value. In this case, we're looking at 6 observations that are both, these observations, or the sample of observations, is both accurate and precise. The mean value of these observations is very close to the true mean value, and they are precise because they are close to each other in value. In this example, we see again accuracy because they will average out close to the mean value, but the observations are not close to each other in value, so not precise. Here we see an example where the observations, the sample is precise, but it's not accurate, and in this case, neither accurate nor precise. Now, bullseyes, when we're estimating values, we often are not actually using a target. And so let's look at a different example. In this case, what we're interested in is just estimating the weight of one individual. And this is being done by groups of observers. They're family groups of 3 at the county fair. And so we want to see if different families are more accurate and more precise in their estimates. So let's take a look at this closely. Family group 1 has a very precise estimate. They're close to each other. You can see that by having this small standard deviation. Family group 2 has a sample of their estimates is less precise, and family group 3 is even less precise than that. Now, normally we don't know what the mean of the actual true mean is. In this case, we're able to weigh this goose and we know that its true weight is 25 pounds. So in this case, we would know that the family groups 1 and 2 were equally accurate, but family group 1 is far more precise. We also can see that family group 3 is neither accurate nor precise. So in data gathering activities, we strive for both accuracy and precision. But remember that the population parameters are unknown. They are fixed, but we very rarely will know what those population parameters actually are. We use sample statistics to estimate the population parameters, and the difference between the sample statistic and the true population parameter is referred to as error. There are two types of error. Sampling error, also called random error, arises strictly due to chance. This is due to measuring a subset of the population. They tend to cancel out as the sample size increases, and the amount of sampling error can be estimated statistically. Sampling error affects precision of our estimate. Non-sampling error is systematic error. It's related to faulty equipment, technique, or human errors, and it introduces bias, either consistent over or underestimation of values. It is not detectable statistically, and non-sampling error affects the accuracy of our estimate. So let's look a little closer at examples of sampling error. In this case, we're looking at 3 schematic diagrams of the same population. The true population size is 400 plants, and they are organized in terms of density of a true mean density of 4 plants per quadrat. In this case, we're using 10 quadrats to estimate the density. And what we see here in these 3 samples is just by the random location of the quadrats, we can result in very different estimates of the mean and also different precision of our estimates. These differences are due strictly to sampling error, just chance location of those quadrats. Non-sampling error has a number of sources. Potential sources of non-sampling error include subjective estimates, the non-random location of sampling units, improper training, inconsistent techniques among observers, misidentification of species, faulty or broken equipment, poor data recording in the field, or data entry errors. So basically, mistakes are the sources, one of the main sources of non-sampling error. And the problem with non-sampling error is that it introduces bias into our estimates. And remember, it's not detectable statistically. We want to reduce non-sampling error, and we can take a number of actions to do this. Calibrate our equipment, clearly define the protocol for sampling, carefully train observers, and randomize to avoid subjectivity. We assume that in the absence of bias, precision leads to accuracy. So to review, we sample to better describe populations. Sample statistics estimate population parameters, and we most often focus on descriptions of central tendency and dispersion or variability. We strive in our sampling for accuracy and precision. Sampling error is due to chance and affects precision of the estimate, whereas non-sampling errors introduce bias and affect the accuracy of our estimates. In the absence of bias, we assume that precision leads to accuracy.